Okay, so I think we are live again. Here we go with take 2. So this recording will replace the second half of part 1. So I titled it 1.5 for the 13 slides from swallowing through stomach that you can only see the red dot moving but not hear my voice. Okay, so I think this part worked on the other video, but I figured I'd start with deglutition from the beginning again. Okay, so here's your bolus of food. So you've chewed it up to a point where you feel like swallowing. At the point when you decide to do this, your tongue is going to push up on the soft palate. The uvula will push up, closing the nasal passages. At the same time, this is the epiglottis. This will fold down and close off your trachea opening. All right, and then the upper esophageal sphincter should open. So here we are, this has all occurred. Notice the uvula is here, blocking the nasal passages, the tongue is pushed up. The epiglottis is closing the airway down below, and that bolus of food is passing into the esophagus as that upper esophageal sphincter is relaxed. So now the sphincter closes as the bolus of food moves past, and from here on out, the circularly arranged muscle will contract in succession, slowly pushing this bolus of food down by peristalsis towards the stomach. So pretty much your esophagus is literally just moving that bolus of food from point A to point B. Okay, so it's moving down. Once it gets down towards the bottom here, the gastroesophageal sphincter is closed at this point. Another way to write this is lower esophageal sphincter. I'll type that for you. So aka lower esophageal sphincter, there you go, is closed. As that bolus approaches even closer, then this will relax, allowing the bolus of food to pass from the esophagus into the stomach. So here we are, that sphincter is opened, all this muscle up here is relaxed, this one's contracting, and will continue to push that bolus of food into the stomach itself. All right, so now we're to the stomach. The stomach serves a few functions. It, of course, is a main place where mechanical digestion occurs after the mouth. So there's a lot of grinding and mixing of the food stuff that you've swallowed along with the liquid with gastric juice to break up the pieces of food into smaller and smaller pieces. Okay, some of the structural features that we need to be concerned about in your stomach. You have, again, this is a grinding organ. It's trying to smash this food into smaller pieces. So at the muscularis has a modification in that there's a third layer. So you have your longitudinal, circular, and then an oblique layer of muscle. So there's 3 different layers of smooth muscle to facilitate the strong contractile waves, which I'll show you in a minute, that kind of smash the food down against this mucosa here. Okay, notice the mucosa is nice and bumpy. That's the rugae of the mucosa. That texture is created by that muscle layer and the mucosal layer, the muscularis mucosa. And that rough surface helps to further break down the food as it is smashed with the gastric waves here. A few other features here. This is the pylorus region. This is the pyloric sphincter that governs movement from the stomach into the early part of the duodenum of the small intestine. Okay, so again, stomach serves as a mixing chamber. It's also a holding reservoir. Only a little bit of the chyme, which will be produced in the stomach, leaves and goes into the small intestine at a time, allowing for slow exit of the food you've eaten to facilitate further digestion and absorption in the small intestine. Okay, so the mucosa of the stomach has some features that you couldn't see in the last picture. There are gastric glands that open into gastric pits. Okay. In those glands, there's a few different types of cells. There's 3 exocrine cells, which are number 1, 2, and 3. You have mucous neck cells that produce mucus. Shocking. It's protective. It protects the stomach lining from the strong acidic environment. Okay. Then you have parietal cells that produce and release. I'll change the color here. Oh, hold on. Bear with me. I don't like the black color, so I'm changing it to blue. Okay. your parietal cells are going to secrete hydrochloric acid. Hope that doesn't fit there. Or HCl. Okay. Chief cells secrete two enzymes, pepsinogen, which is inactive and will turn to pepsin, which is a proteolytic enzyme that breaks down proteins, and then gastric lipase will be secreted here but not active until the small intestine. G cells are your endocrine cell that secretes the hormone gastrin that helps stimulate a lot of different parts of gastric activity in the stomach. Okay, so total secretions, your mucosal layer secretes 2 to 3 liters of gastric juice, including the things that we just talked about, into the lumen of the stomach each day. And again, the muscularis has a third inner oblique layer of smooth muscle. Okay, so let's look at the stomach. So here is the lumen. This is where the chyme or food stuff would be. Here are the gastric pits, these little holes that open into gastric glands. So those different types of cells we talked about are along these gland lines, each individual cell. Notice there's the blue ones, etc., are all the G cell, etc., that we saw on the last slide. Notice here so this is the mucosal layer here. Here's the little layer of muscle, the muscularis mucosae, right there. That is causing the surface of the mucosa to have that rugae, that rough appearance. Here's the submucosa, which is your standard blood vessels, etc. And here you see the muscularis with the 3 different layers of smooth muscle. Okay, so not much absorption occurs in the stomach. There is some, okay, but not too much. So a little bit of nutrients, some ions, some of the shorter fatty acid chains that aren't very long. Some drugs like alcohol and aspirin are absorbed this way, and of course some water. Other than that, the rest of absorption occurs in the small intestine. Okay, so now to mechanical digestion. So your stomach has what are called mixing waves. They start as very gentle, rippling peristaltic movements that stop at the very top of the stomach and move all the way down towards the pylorus every 15 to 25 seconds. This will continue and get stronger and stronger with time, creating what is called chyme. Okay? And chyme is a soupy mixture, soupy liquid, which is a mixture of course, the food and gastric juice. Okay, as mixing continues and the food is breaking down and broken down to create the chyme, the mixing becomes stronger and the waves become more forceful. At some point with each wave, once chyme is formed, about 3 milliliters of chyme is pushed out the pyloric sphincter into the duodenum with each new wave. At this point, if you want to pause this and go look, I'm going to do my best to post some videos showing you what these waves look like in the stomach. Okay, here is just a picture of it. So the gastric waves or peristaltic waves will start here and will flow down, okay? Again, smushing the food particles with the gastric juice. This is pyloric valves closed, pushing against the rough rugae, again to grind and smash that food. At some point when chyme is there, the waves get stronger and stronger, causing about 3 milliliters to come out. And sometimes will come back in. So sometimes there's a movement of chyme out and back in of the stomach. Okay, so here's the video that will attach for you later. So this is propulsion, moving forward, grinding. This is closed, and we're pushing the food stuff against the rugae. Retropulsion is when stuff goes out and back into the stomach. Okay, so now we're on to chemical digestion. So by now you all should know that your stomach has a very low pH for a couple of reasons. So first, how do they generate the pH? So from the parietal cell, this is the parietal cell here. This is ECF. This is the lumen of the stomach, or actually the gastric gland, where this will be secreted and eventually go up into the lumen of the stomach itself. Okay, so we move chloride is moved from the blood and secreted out of the cell into the lumen of the gland, and bicarbonate is as well. So how does this actually, well, bicarbonate comes in. So CO2 actually comes in to the cells by diffusion, combines with water, where the equation that you know and love, that will turn into carbonic acid, which can then be converted into a proton and a bicarbonate. So the proton is pumped out by this primary active transporter, the potassium proton ATPase. Okay, so ATP is broken and a hydrogen ion is pushed out here, and a potassium is pushed back into the cell. Okay, leaving a bicarb behind that will be reabsorbed back to blood, causing this alkaline tides, because now we've taken CO2 and we've changed it from a weak acid, combining it with water. We've taken the hydrogen ion and shoved it out in the stomach to make the H. This is hydrochloric acid. And now it's more alkaline as it's going up because there's a higher concentration of bicarbonate, which is a base. Okay, so the parietal cells secrete hydrogen ions, like I said, and chloride separately, but together that equals HCl, hydrochloric acid. Okay, this acid kills many microbes. Okay, so that's really important. That's why in your upper respiratory tract, the cilia moves your trapped particles of mucus down to the pharynx and the lower up, so you can swallow it, and your stomach acid can take care of many of the microbes that have been inhaled in some way or form, okay, or eaten in this case. It also denatures proteins. The protein denaturation is basically the idea that a protein is now unfolded. So if you recall from A&PII or Bio 1, proteins have a very specific 3D shape called a tertiary shape that is important for their function, for who they interact with. If that shape changes in any way and they start to unfold, that protein loses its function. Here, we're not so worried about losing its function because we're eating food that we want to denature the proteins in. So the protein unfolds from this little 3D ball into more of a long line of amino acids, making it easier for proteases such as pepsin to go in there and start cutting it up in pieces. Okay. This whole process of secretion of hydrochloric acid is stimulated by the hormone gastrin that we mentioned a minute ago, and the parasympathetic nervous system, and the acetylcholine released at this effector. Okay, so chemical digestion continued. So protein digestion of chemical digestion begins in the stomach. So pepsin is the only proteolytic enzyme. Proteolytic means breaking of proteins. It's designed to work at the pH of 2, which is handy because that's the exact middle of the range of the stomach pH. It's secreted as the inactive form pepsinogen by the chief cells, and pepsinogen is activated by the low pH. Okay, so now let me ask you, why do you think we have to secrete an inactive form? Why not just make pepsin? It seems like an extra step that's unnecessary, but it's not, of course. Why is it necessary? It is because pepsin is not picky. It'll cut any protein you get near, including the ones inside the cell that makes the pepsin. So that's a bad idea. So pepsinogen doesn't do anything, and it's in the wrong shape with extra pieces of protein there. As soon as it reaches the low pH, part of it's cleaved off, and now it is an active protease. Okay, there is no chemical digestion of carbohydrates. Salivary amylase is now inactivated. It's actually denaturing, and pepsin's doing its thing, chopping it up in pieces. And carbohydrate digestion does not continue until the small intestine. Okay, now on to lipids. So remember, your tongue has 2 lingual glands. They secrete lingual lipase. That's not active until it gets to the acid here. So a lipase breaks lipids. So it takes triglycerides, which I'm not sure if you know what triglycerides are. So basically you have a glycerol backbone, so 3 carbons, each of whom have a fatty acid on them. So these are 3 fatty acids. Bear with me as I draw with the mouse. Maybe better than when I draw on the actual board. I don't know. You have to tell me that. Okay. So these fatty acids are long hydrocarbon chains. So you basically take, there's a carbon and a carbon and a carbon all the way down. So there's 3 different fatty acids attached to this glycerol backbone. That's a triglyceride, 3 of them there. So it's broken down into fatty acids and diglycerides. So you take a fatty acid off. So basically you can take This whole thing is cleaved off, and then you're left with, this would be the diglyceride here. Okay, so lingual lipase is active here, and gastric lipase is actually secreted by the stomach, but just like lingual lipase, it's not active until it gets to the next place with the proper pH range. So gastric lipase is inactive until it reaches the pH of 5-6 in the duodenum, and then it can do the same thing, breaking down triglycerides to fatty acids and monoglycerides and diglycerides. Here is showing you, this is a gastric gland. There's the pit. So you have the different types of cells that make up the gastric gland. Right now we are focusing on the chief cell that produces pepsinogen. Okay, here's a parietal cell, it's making HCl. So imagine that all of this is actually occurring in the lumen of the stomach. It's not actually occurring next to this gland, it's occurring way up here. Okay, but generally speaking, parietal cells produce hydrochloric acid, so the low pH is there. Chief cells produce pepsinogen, which is a zymogen. It's another way to say an inactive form of an enzyme or protein. The low acid causes cleavage of this orange part of the peptide off. Here's pepsin, that's now an active protein scissors essentially. So here are some of the proteins that you've eaten. Already the low acidic environment has caused these proteins to unfold. This one's not showing the proper shape, but anyway. Then pepsin comes in and can cleave some of the peptide bonds, making partially digested or smaller fragments of peptides. And that's that. Now, wish me luck and pray that I